perfectly in time. I think we can move to the first part of the round table. I think Erwan, you may have a
yes,
there is a slide.
So we share them. So I suppose that this one, yes. So you can see my slide?
Yep.
Yeah, okay. Okay. So maybe we will wait until Damien comes back.
It's well known that the times goes more slowly in Nantes than in other places. In the meanwhile, if there are questions, maybe I don't know. Hélène, is it possible to give the mic to allow people to open the mic if needed, or we shall go through the...
Yes, sure. I can do it if you wish.
Okay.
Maybe it might be easier. Yeah.
So if people want to talk, they have to send their questions around using the question and remark panel.
And then I can allow them to talk.
Okay, so. So.
Damien, are you back?
Looks like I can see him. Okay.
Well, so thanks first to our three speakers for the very nice talks and very different topics and domains, but I think that they are overlapping quite well in the fact that we are now in times where a different approach and converge to the study of different kind of microbiomes. So this is the topic of this session on the round table. So we have on one way different kind of targets, barcodes, genes, or organisms, communities, from collection of samples of or time series, or which are a special way to have collection of samples. We have a lot of resources. We now converge with the viruses, the bacteria, the eukaryotes. So we are starting to gain insights into multi-domains analysis of environments. But still there are many questions that arose: the way that we can sample them, the way that we can use the sequencing data, either the short reads or the coming now, the long reads, either from PacBio or Nanopore, to gain omics data. We also have questions about the integration of other kind of data that can be images, that can be species counts from different observations. So we need to find ways or to have needs to merge those different levels of observations and to connect these different way to apprehend the biological objects, which can be of different kinds. Are there any feelings from the audience, or any insights or any questions that could be raised about this? Rob, sure, yeah.
Yeah, I mean, so I think what I probably presented was, the best bits in terms of not all microbiomes are the same. So to perform assembly on soil is still incredibly difficult because of the complexity of soil microbiomes. And so I think there are still many challenges out there. I think, you know, human microbiome is biased in many different ways. One is the numbers of samples. Secondly, it's quite a simple microbiome. So I think, you know, that there are key challenges there in terms of there still needs to be method development both in the wet lab and then dry lab for tackling these more diverse environments. That's number one. I think accessing the whole repertoire of the microbiome, it presents all sorts of challenges. So viruses, we have so many unknowns out there and they are just so common. We're getting a better handle on the bacterial diversity, but every time you go into anything other than human gut, you're going to find lots of new bacteria. And accessing eukaryotes, you need deep sequencing without doubt, and you need lots of tricks to try and actually assemble and get genomes out. So I know that's only one approach for tackling a microbiome, but I think this sort of shows you that although there is progress being made, that we are far from being able to just take a sample and just immediately know everything that's in it.
Sure. I think that one of the key points is that for the last years, I would say the last 3, 4, maybe 5 years, we're moving from the barcodes or the gene-centric approaches to the organisms. So the mags are, of course, the obvious point there, but this gives us access to the organismal level. So then it's possible, it's more feasible, I would say, to look at the gene content and gene expression in organisms rather than to have a large bunch of genes that we would dig into to try to find biological insights. So I think this is an important step forward that we achieved in the recent years, even though the MAGS approach can still be enhanced, and we can also have better reconstruction and representation of genomes. But this is, I think, a very important point in our field, because we are closer to the biological objects now than we were before. So then we can move. It's easier to move to the communities.
Yes, to some extent, but I would say that there's still, I mean, from that functional perspective, I mean, as we look into these mags, the number of genes we really know the function of is not a large component of that potential coding. And so,
yes, yeah. It seems like we are back to the times when we were sequencing genomes and we were trying to annotate them, where we are still facing the difficulties to what are the functions of those genes, even in a single pure cultured genome approach. But the question is still there.
And to me there was another point that maybe we could raise following the talks of today too. I was very interested by the fact that we more coming back to the need of metadata, in the sense of okay, there is the sequencing technology and there is a new algorithms, but the more it goes and the more we saw the need of beautiful metadata associated to microbiome, because at the end it seems like this is the way that's one way to decipher maybe new species and new because for the first talk of Rafael, it was about the community, but the question of the community and associated to the environment, this is still something that eludes us. The fact about even the microbiome from the skin, to me, we don't have necessarily a lot of metadata, except that you know from where the sample are coming from, and maybe having something on the top should be something interesting. And Sam, also, you were talking about that, about the need of metadata to be able to compare networks and to build network properly. And I was wondering how, especially at EBI, you could connect this with other infrastructures that consider more like Pangea. But to me, the question that could come with this is all metadata are not equal, in the sense that it's not that easy to catch metadata from the gut compared to metadata from the ocean or from the soil or for whatever. So maybe there is an inequality at some point with the different ecosystem that we want to investigate.
Yeah, so to add to that, I also indeed think there's a huge problem in that indeed we lack metadata for a lot of communities, or we don't have it, but we also lack metadata for a lot of microbes that we simply cannot study statistically because they are too rare. So indeed we have these metagenomes for thousands of species, but quite often we cannot actually analyze them just using computational methods. It would be easier if we had metadata to assign them, for example, to a functional guild, because then we could analyze them as a guild and the sum abundance would be interpretable. But at this point, this is still quite hard. And indeed, there are different resources that we can use, but it's not so easy to actually use one of those resources. For example, I've been working extensively with Earth Microbiome Project metadata, which is incredibly helpful. But despite it being one of the most well-curated metadata sets that we have, the amount of ways that people can say coastal microbiome is quite amazing, because you can say coastal, seaside, marine. You really need to dig in to really understand what it means. So I really here see room for indeed more semantic web-based technologies and also natural language processing tools. Because if we use natural language processing, we can use machine learning tools to really understand the context of the communities that we are studying. And right now, to me, this still seems to be missing. Of course, I'd be happy to hear if people are already working on it, because I'd be really interested.
Yeah, Rob?
Rob?
Yeah, I mean,
because I know that you, yeah.
Yeah, I can comment on the metadata a little bit. It's the thing that always comes back, and there are numerous competing things going on in terms of metadata capture. So, I was in a meeting with the UK Research Council that deals with the environment the other day, and the problem what we face is that some of the environmental sciences are going more to omics techniques, but they already have infrastructures for capturing environmental metadata. We don't want to duplicate that effort, but we need to be able to, from an omics person, I need to be able to talk the same language. And I think that there's this notion of different groups still talk different languages about the metadata. And I think that was just evidenced by what Sam mentioned about there are so many different ways to say something that is coastal. It's a constant problem, but if we, the issue is the more requirements you put on sequence submission, the more frustrated people get in the fact that then they don't submit their sequences. And it's a balancing act, and it's a balancing act of where this data goes, whether it goes on the sequence record, whether it goes with the biosample record, or whether it then goes to a third-party resource. Damien, you mentioned Pangea as being another example of where there. But I think all of those are solutions, but it's how we naturally connect across those different resources for storing the metadata. But it is a fundamental problem, and it's not one that I see being solved in the near future. There's also, and I think the best way of encouraging good metadata is showcase projects. So Tara, I mean, I know there's a number of people who are associated with Tara, but Tara gets used a lot because it has such rich metadata. Everyone knows where the samples have come from, you know, all of the environmental, large fraction of environmental factors around them. And often you can get even more if you're willing to make that one step away from the sequence record to the Pangea. So, you know, as the showcase studies demonstrate the richness of the metadata, then my hope is that people will adopt richer metadata sort of reporting. There's also a point on the experimental side of knowing that you need to capture all of these in the first place. But I still have a personal opinion that some people withhold metadata such that they can reuse their data sets for longer, let's just say.
And one of the difficulties of those metadata is that from environmental samples, you cannot go back and complete the data. So once the sample is made, what you missed is difficult to.
But the same is for human. The same is for human. So for example, we looked at trying to sort of look at microbiomes of type 2 diabetes, but type 2 diabetes is a massive range of disease. And so you actually need to capture far more. You not just type 2 diabetes. You're lucky if it says that, but you need to know at what sort of stage, what's their blood glucose level, etc. And for those, you can't go back either.
Yeah. We can also consider the human as an environment. It's the same. And it's strange or fun or sad that the more data we collect, we have, and the more data we want afterward. So it's never-ending story.
There was actually
any kind of Sam, yeah.
Oh yeah. That was actually what I was about to say. The first thing I did after I finished my 60 study network analysis was see if I could get more metadata, because I couldn't separate the samples well enough based on the conditions that they came from. Also, when you have I mean, it's not just that. Like, I think we understand that if you have type 2 diabetes, then you need to measure things like, well, blood sugar is an obvious one, but also other blood metabolomic data. But simple characteristics like assigning the data set the label human, these things also are already quite often not done. So you have a study, for example, on an ocean mesocosm, but that study will not have the keyword ocean added. So if you're just going to search for ocean data sets, you might not necessarily find it. So it's not even necessarily measuring all these different variables, but simply the metadata linking these data sets to the ecosystems that they came from is already quite challenging sometimes. And you need to do it manually using the study in one hand and Excel file in the other hand.
If you can get to the Excel. I mean, there's a limitation in terms of when we were doing the human data sets, 13,000 data sets to actually capture the metadata. This just didn't scale. We could go for the large one, but there's a long tail that we just had to just ignore.
But maybe that could be a good topic, maybe to introduce the second phase of the round table, is the Elixir initiative on microbiomes. So maybe this is a place where this can be more formally discussed and designed, common ways to, or standardized way to collect and to distribute the metadata associated with any kind of samples. That's a way to reuse them, to have the most reusability of the data. Maybe Erwan, you want to introduce this, and then we can have comments from Rob, who is one of the leader, or maybe Rob.
Yeah, I don't know, in fact, because I think that since Rob is there, I think that it's the most. Yes,
indeed.
Yes. So just to introduce the stuff. So the idea that we were both participating with Eric to the marine metagenomics community within the Elixir. And I think that most of you already know how Elixir is organized. So it's organized around platforms. So there is data platform, tools platform, compute platform, interoperability and training platforms. We provide services for different community, and the marine metagenomics community is one of the, let's say, 8, but I think there is more than 8 because there is specific community for human data. So it's maybe more around 10 community, 10 or 11 community. So the marine community have done plenty, there is plenty of achievement during especially during the Elixir Accelerate program. And at the end of the Elixir Accelerate program, there is an opportunity, in fact, to move, in fact, from a marine metagenomic community to a more wide microbial community. And maybe I can let you the floor, Rob, to introduce this community, if it's still an open idea.
Yeah, so there's lots of reasons for the diversification from a marine specific community to a more general community. And I'll just sort of give some sort of overview thoughts on why that's come about. The first is that many of the approaches and many of the issues are common regardless of the biome of interest. There's also the fact that on Owen's previous slide, he showed some of the other communities like proteomics or metabolomics. They are general communities, but they are much bigger. With size comes both positive and negative aspects, but one of the feelings was that actually a lot of what we do is perfectly transferable. So to generalize the marine community, because that was small initially to fit a specific Elixir Accelerate grant, the plan is to now generalize this to focus specifically on sequence-derived information from the microbiome and to generalize this. What we want to try and do is use some of the aspects we've established with the marine microbiome, like standardization of workflows, the establishment of resources, and understand how they can be generalized to a broader community. One of the key challenges of establishing that community is to prevent it getting too dominated by people interested in human microbiome, just because there's more researchers across Europe in that area. We believe that there will be mechanisms put in place, such as an independent steering board that will help us balance that. But we really want to go across the entire spectrum of the microbial composition and really start aiding people to meet the challenges regardless of the microbiome. This is a little bit caught up due to COVID and all of our additional pressures on our time. So this is slightly being delayed from the initial thoughts, but this has been significant. This has been endorsed by the heads of nodes and the Elixir Scientific Advisory Board that this is definitely a sensible way forward. So I guess there's a, from someone who's trying to establish that, is for people in France to connect with their heads of node and their representatives in the microbiome community, i.e. Eric and Erwin, and really start up a discussion about what are your informatics needs, what infrastructures do you need, and to start engaging in with this community, such that we can start making sure that we meet the demands for the infrastructure across Europe.
Okay, thanks. So the idea of this slide was more to give you information, in fact, about this initiative of creating this community, to give you an opportunity to think about that and to come back to us if you have more questions during the time that this community will be built. I think during these next months, probably, There is a white paper that has to be right and submitted. There is stuff like that that are still ongoing. But it's just to give the information to the whole community, or at least to the 50 persons that are still in the symposium, about this initiative, and think about it, and think about the tools or the needs, as Rob said, but also the tools that they could provide also, or algorithms they are working on that could be used to help this larger community who works on microbiome genomics.
So this covers soil, water, different kind of water, air. We try to keep apart the human body related biomes so far, but all the other ones are of course welcome to join us, to jump in, and to participate in the building of this community, because it will have some impact in the next years. Most of the EU funded program will be associated with, or will be connected with the Elixir infrastructure. So this is important also to get involved and to be part of this. So this is also a call for participants to join us, and we believe that this was the good opportunity, this symposium, this JOBIM meeting, to pass the message and pass it again to the people that you know there, because it's important also for us to be in this story.
And Eric, just to clarify, human will not be excluded, but it will be made sure that it's not dominating. So we'll be keeping a balanced approach. I think one of the things to be thinking about is, if you've got a really good tool and you want to showcase it, this is a really good way of trying to engage with people like myself who provide services. If collectively we can demonstrate that this is really important for the community and should be established in key services, that's what we've done. So we've taken both tools from the community and resources, so reference databases, and put those in Magnify. So this is, if you've got something that you think will improve, then that's one area of engagement. Another one is finding common topics. We've heard about this metadata issue. There are so many different aspects of metadata. One of the simple things we've been looking at is, can we, quite often people don't even report in a meta barcoding which of the variable region they've actually amplified. We can assert that sometimes just purely from the weight of data. Can't always do it if you've used multiple amplified regions, but a lot of the time we can assert that. So we're now looking at ways of capturing that information and then putting it back into the record. But that's just one metadata field. If you've got smart ways of thinking about how to infer metadata, whether that's through text mining or using machine learning to contextualize data sets where there's no metadata, with those that do have metadata, to try and say, we're pretty sure this is a marine data set rather than a terrestrial data set, that's at least something is better than nothing. And so we want to try and capture these key challenges that people are facing and gather a corpus of people who are interested in addressing that problem.
Exactly, yeah. Yeah?
Yeah.
Someone else wanted to add something?
So,
nope. I think it's time. So it's 6. It's already more than 6 here in France. 6. Yeah. So maybe it's time to wrap up. And thank you very much. That was very interesting, very cool.
So thanks a lot
to all the participants. Yes. That one.
Yeah. It was to thanks also Sam, Rob, and Raffaele for their talk, and thanks all the people who stay here for the roundtable, and hope that we will have a more more exchanges about the different questions that we raised during this roundtable, and this opportunity opened by this new community in the next few months or years.
Yeah, feel free to contact us to get on board this emerging community. This is important. Yeah.
Okay.
All right. Thank you.
Thank you.
Thank you.
Bye.
Thank you.
Bye.
See you.
Thanks you all. Bye bye.